Hi, so I'm going to tell you a bit about our new paper out in the Journal of Neuroscience. It's called No Evidence for Entrainment, Endogenous Gamma Oscillations and Rhythmic Flicker Responses Coexist in Visual Cortex. So the main idea of this paper was to ask, can we entrain and manipulate gamma oscillations in the visual system in order to understand what they do from a functional point of view. So why gamma oscillations? Why might they be important and interesting? Well, first of all, we know that if neurons that synchronize in a gamma band, they have a strong impact on downstream targets. So this is illustrated here by the top figure. So the red lines here indicate neuronal firing. Then the postsynaptic potentials of these neurons are going to add up if there's synchronization in a gamma band, and thus make the downstream neuron fire. This is less so the case for synchronization in the alpha band because it's less tight from a temporal point of view. Another motivation for studying gamma oscillations is that they have been proposed to be involved in binding. So consider this picture of a woman and a cat. Some neurons are involved in representing the woman and some the cat, but how does downstream brain regions sort out what neurons correspond to coding for the woman versus the cat? Well, the idea is that synchronization in a gamma band would reflect the formation of an assembly. That could be the woman, and then another group of neurons synchronizing also in a gamma band, but say a slightly different phase of frequency would represent the cat, right? So it has been proposed that different objects sort of bound together, the features are bound together by means of gamma synchronization. The third idea is that communication between different brain regions is also achieved by synchronization in the gamma band. So basically, two brain regions that synchronize at gamma frequency would also be exchanging information. So there's plenty of good reasons for studying gamma band oscillations, and we here try to manipulate those by entrainment. So what do I mean by entrainment? So consider this example where there is a girl on a swing and a boy pushing her. So this girl on a swing would of course have an endogenous oscillation associated with the swinging, a so-called resonance frequency. Now if the boy is pushing her at the same frequency, the swings will get larger and larger. So this is exactly what we think about when we say entrainment, and what we are trying to do in this study is have the endogenous oscillations being the gamma oscillations, and then we'll drive those by an external flicker, and hopefully we'll be able to hijack the dynamics and boost these oscillations. So to study this, we used MEG. MEG is great because it allows us to measure the electrophysiological activity from the human brain, but also to localize where in the brain the activity is being generated. Furthermore, we made use of this ProPix projector. It has a refresh rate of 1440 Hz, so it allows us to drive the individual pixels, if you will, in the visual display at arbitrary frequencies. So it allows us basically to flicker different parts of the scene at particular frequencies. So the first part of the study just involved having a gray patch in the center of the screen flickering a given frequency, and then we measured the response. It should be mentioned that when you drive at frequencies above 50 Hz, people don't perceive this flicker because it is above the flicker fusion frequency. So so when we do this experiment simply by flickering the patch in the center of the screen, we do get a sizable response. So here's an example for two subjects. Each peak reflects the frequency at the respective stimulation frequency. So as you should observe is that we get a several-fold response, response with respect to the baseline, right? So there's really a sizable signal hammering through when we do this flicker. The topography of this is over the back of the head, the visual cortex, as you would expect. When we look at the grand average over multiple subjects, we see a systematic decrease in the flicker response, and at 80 Hz we don't see a response any further, right? So this means we can drive the brain with visual flicker up to about 80 Hz. So the next paradigm is somehow more interesting, because what we do here is that we make the brain oscillate at endogenously at the gamma frequency. So we do this by having these concentric gratings moving towards the fixation cross, and it has been shown in many studies that that induces a very strong gamma band oscillation. In this case, a fourfold increase with respect to the baseline. So this red color here indicate the endogenous gamma oscillations, in this case at 58 Hz for this particular subject. So the important thing here is that different participants have different endogenous gamma frequencies, right? So the second participant has a gamma frequency at 74 Hz. And again we see these gamma oscillations over the back of the head. So this study or paradigm was designed such that we had the movement gratings, and then on top of those 2 seconds later we put on a flicker. So the interesting question here is, if we now sort of flicker the system at the endogenous gamma frequency, can we boost the endogenous gamma oscillations? What you see here is the power spectra, or the calculated for the different frequency ranges, or the changes in power for the different frequency ranges. And what you should appreciate is that there's this systematic drop in signal with respect to the stimulation frequency. When we both have the flicker and the gratings on at the same time, we get a stronger response as compared to the flicker. So what we now did was to go in and shift these spectra with respect to the endogenous gamma oscillations. So if one participant had 68 Hz as the endogenous gamma oscillations, we shifted these 68 Hz to 0. And if somebody had 76 Hz as endogenous gamma frequency, we shifted the spectra accordingly. So you see the result here to the right. And what you should then note here is the blue line being most interesting, because there we have the flicker together with the gratings. And what you should note is that at around the endogenous gamma oscillations, here denoted by 0 Hz, we do not see a boost in a gamma band oscillations, right? This is what we had expected had there been entrainment, but we do not see that. So basically there's no evidence at first here for entrainment. So what might be going on? So let's just consider the time frequency representations of driving flicker frequencies, different flicker frequencies. So this here is a bit complicated to look at, so I'll just zoom in at one plot. And what you should appreciate here is that from time 1 to 2 we have the movement gratings, and at time 2 to 4 we have the flicker together with the movement gratings, and then after time 4 we have the movement gratings only. And what you should appreciate is that the flicker response and the endogenous gamma oscillations, they coexist. Indeed, when we look at this example here for one participant for different flicker frequencies, we typically always see the flicker response and endogenous gamma oscillations. We never really see that these flicker stimuli are hijacking the gamma oscillations. So it's not like when we, for instance, drive at 52 Hz, we are able to sort of steal the gamma band activity as one would expect if there had been entrainment. We can also go in and align these individual spectra here according to the endogenous gamma oscillations, and then do the grand average, and this is what you see here. So in this example here we are 4 Hz below the endogenous gamma frequency with respect to the flicker response. And again, there's no evidence that we steal the dynamics. Since we're doing MEG, we can also localize the sources of the gamma oscillations, as you see here to the left, and the flicker response as you see to the right. So now, if you compare the peaks of these sources, what we see is that they indeed are at different locations. There's some overlap, right? But the peaks are different. So this here also suggests that different populations are involved in generating the gamma oscillations and the flicker response. So I'm going to summarize here. So basically, the core conclusion is that the gamma oscillations and the frequency tagging response coexist. In other words, there's no evidence that we can entrain the gamma oscillations by this visual flicker. And at first we were somehow disappointed about this because it is indeed a null finding, and we cannot go in and manipulate the gamma oscillations as we had hoped to do. But then again, it's also interesting because it does demonstrate that these gamma oscillations in the human visual system are very robust. And what we now think is going on is that we cannot entrain these gamma oscillations because the feed-forward projections in the brain are done by excitatory connections. And there's work being done using optogenetics that has demonstrated that in order to entrain gamma oscillations, you have to drive the inhibitory neurons, not the excitatory neurons. And this is simply what we are not doing by the visual flicker. So I should also say we have another study very much speaking to the same conclusion. So it's a study by Alexander Sigalov out in PLOS Computational Biology, and there we did a broadband flicker input, and then we asked, is there amplification in the gamma band? And the answer is no. So again, this is also further confirmation that it seems not possible to entrain endogenous gamma oscillations. I will end here and acknowledge the different people involved in this study, in particular Duecker, who had the basic idea to the study and did the data collection, design and analysis and so forth. So the findings here is really to her credit. And then we had Gutteling and Herrmann also consulting the study here. Thank you very much for your attention.